I would like to thank you. So thank you very much, Clarice and colleagues, for being here. I'm sorry, apologize for speaking to you online from the distance, but I'm sure this is going to be fun. And we'll talk today about cracking codes, cracking redox codes, and I'll try to, in 20 minutes, to explain to you what that means. I belong to the Vascular Biology Laboratory in the Heart Institute of São Paulo, University of São Paulo. And this is my most important slide. All the collaborators that are the postdocs, the associated investigators, PhD students, undergrads, research assistants, collaborators, and past members who helped us to actually build a code and try to crack this redox code, which is, and here I will introduce you to the main actors, which are the reactive oxygen species, which many of you know as free radicals, but not all of them are free radicals. Some of them are indeed free radicals, which means they have an unpaired electron on their outer layer, like superoxide, hydroxyl radical, peroxyl, alkoxyl, nitrogen oxides. But there are several other non-radicals, like hydrogen peroxide, hypochlorous acid, ozone, and singlet oxygen, which is an excited species, very important for potential quantum biology effects. And these actors actually combine on the biological scenario such that they are produced during several metabolic events and also during disease like atherosclerosis, diabetes, cancer, and they are not produced by accident. Very rarely they are produced by accident. For example, an event of radiation exposure or drug exposure, but most of the time they are produced normally by enzymes like NADPH oxidases, NO synthase uncoupled, xanthine oxidase, cytochromes, etc., and especially mitochondria, of course, which produce mainly superoxide. Superoxide can dismutate itself into hydrogen peroxide. That is facilitated by superoxide dismutase enzyme. Hydrogen peroxide is scavenged by catalase. And as you can see, the rate constant of reaction of SOD is quite fast, but there is still another faster reaction for superoxide, which is a direct reaction with nitric oxide, which is a free radical generated by vascular cells and macrophages and neutrophils and neurons. And the product is a series of nitrogen and carbon radicals, which have several effects. They are powerful oxidants, and some of them can nitrate amino acids like peroxynitrite, can nitrate tyrosine, and thus produce some effect. So the first thing I want to take home, first message is oxidative stress is not an accident. We need free radicals, and actually we need them to do signaling, what we call redox signaling. So oxidative stress is this imbalance of redox signaling, which is the cellular transduction of signals in which the integrative element involves at one or several steps electron transfer reactions or transfer of reducing equivalents directly or indirectly related to intermediate reactive oxygen species. The particular feature of redox signaling as opposed to general signaling in general is that it depends heavily on chemistry in addition to biology as expected. And in chemistry, we talk a lot about kinetics and thermodynamics, which I'll barely touch today, but it's important. What is the second notion I would like you to take home is that redox effects are expected from proteins in our organism and from lipids. They are not necessarily damaged by the oxygen radical. So That can happen, of course, but I will not talk about this today. Today, I'll talk about proteins that are made on purpose to be modified by redox species, and these contain mainly redox active metals or cysteine thiols, which are the focus of our talk today. Cysteine thiols, a thiol is an SH or sulfide group, which can be reversely modified by a series of reactions. And in being modified, the proteins that contain cysteine residues are turned on or turned off. And the traditional model is that the type of thiol modification and the pKa of thiols, which govern the ionization of thiols and thus their reactivity, govern signaling. This is a rather complicated slide. As you can see, the landscape of thiol modification. So the more ionized here, the thiol, the more reactive and the less the pKa. And basically, there are two big groups of reactions for thiols, which are oxidant reactions and disulfide exchange reactions in the upper part of the slide. The oxidant reactions can form sulfanic, sulfinic, and sulfonic acids, but mainly everything converges to disulfides. So I think it's easy to imagine that if you have a thiol that's reduced and then it oxidizes to a disulfide, then the protein changes form and it's turned on or off, and our receptor is changed from an inactive state to an active state, and that can vary. Generally, oxidation activates, for example, kinases and inactivates phosphatases. That's a general paradigm, but there are several exceptions. There are other modifications of thiols, which we'll not talk today. Nitrosothiols, direct formation of a thiol radical, and more recently, persulfidation by hydrogen sulfide has been a very important new addition to the many existing thiol reactions. So generally, to sum up what I have said so far, oxidative stress is a disruption of redox signaling equilibrium, and oxidative stress does not necessarily involve molecular damage. Several reactive oxygen species are generated endogenously by enzymatic mechanisms and may have physiological or protective functions, as well as damaging functions, of course. Cysteine thiols are major redox targets in signaling proteins, peptides, and small compounds. What I'll focus from now on is that the thiol disulfide equilibrium involves several intermediate forms that establish a thiol code based on the type of thiol modification, as you saw on the previous slide, and pKa-dependent reactivity. However, we have a problem here, which is the kinetics of thiol oxidation by reactive oxygen species is very slow. So thinking that hydrogen peroxide can directly modify thiols is probably limited. Thus, we have to imagine that several professional thiol proteins, such as thiol oxide reductases, play major roles as redox sensors and relay intermediates. For example, peroxyreductants, which react very fast with hydrogen peroxide, can be oxidized and transmit this disulfide to other proteins. That's what we call relay oxidation. In other cases, as we'll see, proteins are not as clearly a redox relay intermediate, but they are also contributing to modify targets in proteins. Another interesting case is the case of allosteric disulfides, which are disulfides that form in a portion of protein that's not necessarily catalytic. But they modify the conformation of the protein to allow modulation of protein function. So as you can see, oxidative stress is far from being an accident, but it exists on purpose, and they constitute a true redox code and a very important, let's call it subcode of this redox code is the tile code. So let's talk a little bit more about the tile code and talk about the things that we study at our group. Mainly, the idea of tile code is that redox signaling requires adapters. So you have sensors like peroxyredoxins. You have complexes that generate enzymatically reactive oxygen species on purpose. They are regulated to do that. You have upstream factors that turn all this cascade on, and you have the target here that is modified in several ways by these proteins. One protein that our group has been studying is protein disulfide isomerase, or PDI, which belongs to a large family, and it's a very interesting adapter, and we have studied the PDI code now for many years, and we believe it's a very peculiar code for redox cell signaling. Here is PDI. It's a protein that belongs to the thioredoxin superfamily of proteins. They have 4 thioredoxin domains, A, B, B prime, and A prime. And the A and A prime domains have 2 cysteines each, which are cysteine, glycine, histidine, cysteine. So that is a dithiol motif. And PDI is a very mobile protein, as you can see here on the right-hand portion of the slide. The green part is oxidized PDI, and the purple is reduced PDI. So there's a lot of mobility, which is given by this X linker between the B prime and A prime domains. This mobility is very important to allow PDI to bind to a series of proteins and exert its main function, which is oxidative folding in the endoplasmic reticulum. We'll come back to that. So this is the main function of PDI, as exemplified by the family prototype PDI-A1. So here is a correctly folded protein substrate, which can be achieved in 2 ways. First, PDI may take a reduced unfolded substrate and oxidize it correctly. Second, PDI may take a misfolded substrate, which has disulfides in the wrong position, forms a mixed disulfide, and then yields the correct disulfide. In this process, PDI is reduced and it needs to be reoxidized by this oxidase, which we will not talk about today. The message I would like you to keep from this slide is that PDI is a very important introducer of disulfide bonds, and as such, it's a redox chaperone. And disulfide bond introduction is a very physical-chemical process, actually, in which 2 things happen when a protein folds. One is to remove water from inside the protein. So folding a protein is essentially removing all the water inside it. Second, you decrease the energy, the free energy of this protein to the lowest possible level, and that is probably the native state of this protein, the functional state of this protein. So folding the protein is an extremely important function of PDI. So PDI is a vital protein. There are several other proteins that help folding proteins, many chaperones in the cytosol, for example, heat shock proteins. But in the endoplasmic reticulum, in which many large proteins exist, PDI is the main catalyst. So the classical functions of PDI are protein folding, disulfide introduction, proteostasis, which is protein homeostasis, protection against endoplasmic reticulum stress, cell survival, and there is a pool of PDI in the cell surface involved in thrombosis, platelet aggregation, virus internalization, and cell adhesion. So PDI does a little bit more than only folding proteins in the ER, which is already a very important function, but it is also involved in functions at the cell surface, and our group has described several other new functions of PDI, such as NOx and ADPH oxidase regulation, cytoskeletal organization, cell migration, vascular remodeling, and here are a series of reviews that we have written over these years that summarize these effects. I'll show you only a few examples showing, for example, PDI outside of the endoplasmic reticulum. Here you can see that one portion of PDI migrates in these vesicles together with actin. So PDI help organize actin, cytoskeleton, and when you stretch cells, PDI migrate to membrane ruffle, as you can see here. So these are smooth muscle cells, and stretching is a very physiological effect of smooth muscle cells, which are made to contract and to relax. A second function that's very cool, and this is a collaboration with Adriano Alencar from the Physics Institute of University of São Paulo, is the polarized distribution of adhesion forces in cells. And as you can see here, cells exert an organized scheme of force, and this is traction force microscopy, which measures the adhesion of cells to extracellular matrix. So here is the heat map. So we can see this cell, for example, it has a gradient of force. So it has more adhesion force in the upper portion and lower adhesion force here in the inferior portion, which means these cells are migrate upwards. But when you add PDI, when you neutralize PDI, you see that this gradient is lost. There is some force, although lower, the vector force is lower, but these cells cannot really polarize the cell. And we are now studying why this occurs. We believe there are several targets involved in this process. And as you see here, the net contractile moment of these cells is decreased. So these cells cannot organize force distribution. Actually, they look like they are drunk. They cannot migrate on a straight direction. So the noise of migration is significantly increased by PDI neutralization. So we believe that PDI is a noise suppressor in cells with their function. So summarizing the effect of PDI, PDI effect of associated cytoskeletal regulation. PDI is essentially required for vascular cell migration, and at least the cell surface pool of PDI governs cytoskeletal organization to minimize the noise of remodeling and support migration persistence. And final two slides I'd like to show you are translational implications, because this is a translational function session, and we have been studying how does effect correlates with some diseases. One disease that is highly dictated by mechanical factors is aortic aneurysm and dissection. And here you can, this is a very, very summarized finding of this paper. You can see that mice that overexpress PDI, when exposed to, mice that overexpress PDI, they survive more than wild-type mice when exposed to an agent that promotes aortic aneurysm and dissection. And we know that PDI produces more. And when you inhibit PDI, here in red, the mortality is enhanced because there is an enhanced rate of dissection. So PDI promotes the stability of the aorta during mechanical stimulus. And aortic dissection is very much related to calcification, and here is only one slide to give you a flavor of this paper that PDI overexpression in a transgenic model protects against calcification. Here is a fluorescent indicator of calcification, which is decreased in those transgenics exposed to vitamin D. And here is a quantitative summary. You have more calcification in wild-type mice exposed to vitamin D, but much less in transgenic mice exposed to vitamin D. So these are two examples. Of course, I didn't have time to make all the mechanistic connections, which we still know very little. But I would like to finish with this slide, trying to speculate how redox biology can meet quantum biology. First of all, they are really cool, both. Second, there is a hypothesis that the signal transduction landscape might involve an energetic and entropy fluctuation analogous to those of protein folding. For example, with PDI, you can see that. That's where redox chaperone might get in, and such energy landscape might involve quantum transitions at some step. I would like to start talking to you about that. Second, there are some other interfaces between redox processes and quantum transitions. For example, excited species such as singlet oxygen. And we can imagine also that the quantum brain of the cell might be a redox-dependent circuit, such as mitochondria or a vesicle in which there is an NADPH oxidase. And finally, we can imagine that we can build quantum sensors. One very difficult thing in redox biology is to measure things, and we can have quantum sensors of essentially everything redox. So with this, I'd like to thank you for your attention, and we can come back in the discussion, and I'm looking forward to the other talks in this session. And thank you again for inviting me.
Francisco, I would like you to, if possible, speculate. So, short background story. A postdoc of ours, Abazout, spent almost 6 months working in the lab of Francisco Laurindo. And I mean, even though we need more controls and so forth, we have seen something very interesting. So the postdoc then, what he did is he incubated cells with weak magnetic fields and looked for things that changed in the cells as they were incubated with weak magnetic fields. And one of the things that Abazout saw, Francisco, was a change in the polymerization rate of microtubules. And I wonder if you could speculate on what you think that is due to.
Yeah, this is an extremely interesting story that I think we should really pursue. My speculations are that there are some magnetic field sensitive elements in the cell, such as, for example, calcium channels. That's one of the possibilities, or other types of ion channels. The second speculation is mitochondria, but above all, I think what this really tells us is that one stimulus that changes the magnetic field can promote widely significant consequences in cells. So there's a very interesting experiment that was actually Abazout showed me that when you have calcium transients in cells, and then you turn your cell phone close to the culture plate, that changes the rate and the intensity of calcium transients in the cell. So that's very indicative that some elements are really made to fill the magnetic field. And so we have a magnetic code in cells, maybe, that is sensitive to these stimuli, which probably intersect with the redox code. It's a question of finding the mechanism, Clarice. I think I would bet my first efforts into calcium channels. I think they are possibly the most important connection between the magnetic field and the effects we saw.
Well, thank you. There's another question coming.
Hi, Francisco. This is Sergio Ferreira. Hi, how are you?
Hi, great. Thank you. How are you?
Yeah, so just a comment on Clarice's point here. Microtubule polymerization is actually regulated by kinases and phosphatases. And if memory serves me, these kinases and phosphatases are all regulated by oxidative stress and by redox signaling. So what I'm thinking is that perhaps by imposing some magnetic field, not only you could change ion channels like you suggested, Chico, but you could also change the balance between kinase and phosphatase activities, which would therefore impact microtubule stabilization and polymerization.
Yeah, that's absolutely true. I think that what I thought was maybe things might begin with the calcium ion, but then very soon, later that changes to a redox imbalance. As I told you, maybe for those of you who are not familiar with the redox stress, most phosphatases are inhibited by oxidation. That's a kind of general rule. So if you think of a protein, which protein is sensitive to oxidative stress? Think of a phosphatase in first place. Most phosphatases are inactivated by oxidation, which is a very physiological mechanism that is not damaged. It's inactivated on purpose so that the signal can go through. Because if the phosphatases are generally much more active than the kinases to turn off the signal. So if the phosphatases are not inactivated, the kinases cannot do their job to phosphorylate the substrate. So I think the message I would like to leave is that there are targets of the cell that are modified on purpose by redox agents, and there can be, of course, several quantum effects and interaction with those stimuli that we saw.
Awesome. Thank you very much, Francisco.